So, good evening, everyone. Honorable Vice Chancellor, Jawaharlal Nehru University, Professor M. Jagdish Kumar, Dr. Theresa and her team from Arizona, and their participants from different parts of India, USA, and Canada. On behalf of Jawaharlal Nehru University, University of Arizona, and USA-NPN, I welcome you all in today's webinar on National Phenology Network, Big Idea, Productivity, and Potential. Friends, spring has just left us, and currently we are with early summer. The spring and early summer is the time for initiation and continuation of phenological activities. However, this period of 2020 will not be remembered for phenological activities, for joy of species, but for coronavirus pandemic. I hope, following the advice of infectious disease experts and governments, you all are sheltering at home, practicing social distancing, and keeping yourself fit. You all are aware that since ancient times, human societies have been using phenological observations in scheduling agricultural activities. People used to link phenological observations with weather conditions to support their decisions. Several traditional communities throughout the world still follow these practices. At present, in the face of climate change scenario, science of phenology has emerged as an important branch of ecology, particularly in assessing the impact of climate change to ecosystems. Therefore, to create phenology database and also to monitor changes in phenological events, especially in the face of climate change, large number of phenological networks have been established globally. One of the highly active network is USA National Phenology Network, which is being led by Theresa. During her talk, Theresa will give some more details about it. If we talk about India, this country is endowed with great diversity of climate and vegetation. Even in Himalayan region, within 100 to 200 kilometer distance, we have ecosystem types ranging from tropical broadleaf forests at the base of mountains to temperate coniferous forests and alpine pastures at higher elevations. Need for a phenological network covering all biogeographical regions of the country is being felt by researchers of the country for quite some time. I hope today's webinar would give some directions to this idea. Dr. Theresa has not only agreed to deliver today's lecture on the subject, but also assured all possible help in initiating Phenology Network in India. With this, once again, I welcome you all to this session. Thank you. Now I'll request Honorable Vice Chancellor, sir, to formally inaugurate the session with his commentary remarks. We see, sir.
Right. Dr. Theresa, Professor Garkoti, and my colleagues from within India and outside India, let me welcome all of you to this very important webinar. While we are all fighting the crisis of human survival, it is both a medical crisis and also an economic crisis. As we are talking today, about 82,000 Indians have contracted this coronavirus, and nearly 2,650 have died. India is adding about 3,900 cases every day, but fortunately, nearly half of them are recovering. And during last 2 months, the steps that India has taken has significantly slowed down the rate at which the coronavirus crisis is spreading. I hope that both India and the rest of the world will come out of this crisis sooner or later. But one interesting thing I have observed during this lockdown period when we meet online for webinars is that the webinars start on time, which is a very welcome thing to happen. Having said that, this webinar is co-hosted by JNU in association with USA-NPN and Arizona State University. As all of you know, Dr. Theresa is the director of USA National Phenology. And in today's webinar, she will discuss with us the idea of USA-NPN right from its conceptualization to its implementation. From an Indian perspective too, phenology is very, very important because India is a huge country with a huge population, and right from Kashmir in the northern part of India to Kanyakumari in the southern part of India, from Kutch in the west of India to Bay of Bengal in east of India, the entire country is broadly divided into 15 agroclimatic zones. And as you know, almost 60% of Indian population, it lives in the rural part of India, and nearly 40% of Indians, they work in agriculture. Therefore, understanding and predicting phenology is very, very important if we want to make any evidence-based decisions in these sectors such as agriculture and forest-related services. So it gives me a great pleasure in welcoming Dr. Theresa to share her experiences with USA-NPN, working in the field of phenology for so many years. I congratulate both Dr. Theresa and Professor Garkoti for getting together and for organizing this webinar. Let me again welcome all of you to this great webinar. Thank you. Thank you very much.
Thank you. Thank you both so much, Dr. Kumar, as well as Dr. Garkoti. Those were wonderful introductions, and I really am very excited to share with you what our experience has been here in the United States in establishing and growing a national phenology network. And we've been at this since 2007, so going on 13 years. And so that has been honestly a long time to really learn a lot and grow a lot of that. And so that is what I will be sharing with you today.
Thank you very much, sir, for your inspiring words. Now, before I invite Dr. Theresa for her talk, it is my pleasure to briefly introduce her to all the participants. Dr. Theresa Crimmins is the director for the USA National Phenology Network and has been a part of the organization since 2007. Hailing from Ohio and Michigan, she received her bachelor's and master's degrees at Western Michigan University and a PhD degree from University of Arizona Tucson. In her role with the network as the director USA-NPN, Theresa leads an amazing team of individuals and works enthusiastically to support the growth and use of phenology data and resource curated by the USA-NPN, involvement in Nature's Netbook, and a broader appreciation of phenology among scientists and non-scientists alike. In addition to her responsibilities, almost full-time responsibilities in USA-NPN, Dr. Theresa also maintains an active research career and has published more than 60 peer-reviewed articles in journals of high repute. Those journals include Geophysical Research Letters, Global Change Biology, PLOS One, and Journal of Ecology, naming some of them. She has appeared in PBS Productions, Sci Girls, and American Spring Life, as well as on NPR and the Weather Channel. She currently serves on the editorial boards of several scientific journals. With this brief introduction, may I now request Theresa to just begin her talk.
Excellent. Thank you so much. Okay, So to just make sure that we're all starting out on the same page, I think everybody on here is well aware of what phenology is. The working definition, the definition that we tend to use at our institution, is that it's referring to the timing of life cycle events in plants and animals that occur repeatedly, so typically from year to year. And so this is things like when different plants are putting out their leaves or flowering, when birds are migrating in the fall or returning in the spring, when eggs of birds or insects are being laid or hatching, and how those different events are tied to drivers, abiotic and biotic drivers. And that's those things. Generally, what we've recognized, what I think what we all know, is that humans generally definitely have a very clear understanding of what phenology is, but may not understand the term. But we're all paying attention all the time to when plants are putting their flowers out, especially in the springtime, and why that might occur. We all generally also have a sense that it tends to vary from year to year, but we don't, as scientists, have a deep understanding of what is driving that, or how those things might change, or what the consequences are. If we can better understand what drives the timing of events in different species, we can use this information in a wide range of applications. For example, if we can understand when particular invasive species are going to be undergoing a life cycle stage where they may be controlled most effectively, then we can anticipate that and hopefully have more effective management. We also can understand, we can also time or plan for harvest activities and things like crops, if we can understand when they are likely to become ripe and ready to be harvested. If we understand when flowers are blooming and when insects are active, we can understand the dynamics of pollination better. And the timing of seasonal events like flowering is actually really important for cultural events as well. Around the world, there are so many festivals that are tied closely to the phenology of a particular species. Here in the United States, it's very popular to celebrate when tulips are flowering in the spring, or when lilacs are in flower. And in many cases, if those events occur early in a year, they don't always actually coincide with the cultural event that was planned, and that actually can be a problem. And so better understanding when these things might be occurring can really help for planning. In addition to the year-to-year variation in phenology, we also are experiencing underlying clear trends in the timing of when things are occurring around the globe. And it's important to understand how these things are changing too, because the phenology in many species is changing and in many others is not changing, or is not changing to the same degree. And that's resulting in implications for things like available food supply and population sizes and ecosystem functioning. So in this example, we have a generalized example where a bird is migrating from the south to its breeding grounds on the left here, and it lays its eggs. And then as the season progresses, those eggs develop and the nestlings hatch. And this particular bird has evolved over millennia, likely, to have its eggs hatch at the time when the food supply for those nestlings is at its peak. And in this case, it's the abundance of a particular caterpillar. What we've begun to document in a lot of systems around the globe is that with warmer temperatures, events like the hatching of those caterpillars is being advanced and is occurring earlier in the season now. However, long-distance migrants, birds that are migrating from the south, are typically triggered by things like sun angle rather than warmth in the spring. And so those events like the migration aren't necessarily advancing and occurring earlier like the abundance of caterpillars are. And so if the birds aren't migrating earlier, or they're migrating earlier but not to the same extent as the advancement in the caterpillar, then we may be setting ourselves up for having the eggs hatch when the caterpillar food abundance has passed its peak. And this kind of situation has actually been documented in a variety of systems already, and just gives it one of many examples of how changing climate conditions are impacting phenology and therefore having significant consequences on ecosystem functioning. So with the recognition that being able to anticipate when phenological events might occur for planning a lot of short-term planning decisions, as well as the fact that phenology is changing in a lot of species, led to the establishment of the USA National Phenology Network back in 2007. We primarily exist to collect, store, and share phenology data and information. So for the most part, we are the network that encourages this data to be collected, and then we make it available through a variety of means. But for the most part, we are making that information available for others to use, to advance science, to advance our fundamental understanding of what drives phenology and how things are changing, to support decisions, also to communicate and connect with folks across the country and around the globe about what phenology is and why it matters and how it's changing and what the consequences might be. And then finally, to create an equitable and inclusive network. Our aim is that everything that we do is of benefit to ideally everyone in our country, regardless of their background, their geography, their race, their political alignment. So how we undertake these activities can be captured in this generalized workflow, where we start on the left with a partner or a data user, someone who is interested in some phenology information, someone who says, I would really like to know more about when a certain thing occurs because that is important to me in my work. We work with them to try and understand to the best of our ability what it is they need. And then that generally leads toward us encouraging more data collection, because typically we need more information, more data to be able to understand the event better. That typically then leads to a greater understanding in the form of a model. So in this case, typically this is, when is something occurring and what is it that drives it? And if we can nail that down, if we're able to identify with a reasonable amount of error what a driver of a phenological event might be, then we can use that to then make forecasts, predictions, maps, and information in the form of various products. And so I will now walk through those different phases and show some of the activities that we have been undertaking at each of these phases. So starting on the partner needs and data collection side of things. One of the very big things that we do that is a lot of effort is to run the program called Nature's Notebook. This is we call it a comprehensive community science program, because it is the collection, it's the program through which we encourage both professional scientists and volunteer scientists to help us collect information on what's happening on the ground over the course of the season from year to year. And so how the system works is that at its core, we've got standardized observation protocols, which tell folks what to observe and how to observe it, basically. And we have those available currently for about 1,400 species of plants and animals. And those protocols were developed in collaboration with scientists to ensure that they actually were going to yield as useful of information as possible. And then we also provide extensive training materials and support for folks when they are signing up and learning how to participate. And the program is available for use as on paper data sheets that can be entered into the computer, as well as on mobile apps. And so you can, once you get everything set up, it's very quick and easy to be able to go out in the field and collect data on your smartphone or your tablet, even if you are not within the data collection network. You can collect if you're outside of the range of Wi-Fi or cell service, it will still log your data on the device and then upload it later. What the protocols look like are a series of yes/no questions. And I provide kind of the full, a peek here into the full descriptions, just to give you a sense of this, since it sounds like you may be interested in implementing something similar there in the Himalayan region and perhaps beyond. And so this is just a quick peek at what one of our data collection protocols look like. This is for the plant wild parsnip, which is shown on the right there. Each time that you're going out and making observations, you're making observations on the same individual plant. In this case, you would probably identify a patch of them since they grow in a clump, but you'd be making observations repeatedly on that same patch of individual plants. And then you're prompted with a series of questions that start with, do you see? And so in the case of this plant, you would first be prompted with, do you see initial growth of leaves? And if you're not sure, then there is a subsequent description that elaborates on what initial growth in leaves look like. And so in this case, you can read it here. It's new growth of the plant is visible after a period of no growth, either from above ground buds with green tips or new green on white shoots, so on and so forth. And so the intent there is that we provide enough information that an observer, even if they're not a botanist, they don't have background, strong background in plants at all, that they could still read this and look at their plant and make a judgment with a high degree of accuracy as to whether there really is initial growth or not. And then if you end up answering yes to a question, there are frequently what we call intensity or abundance follow-up question. So for this plant, after you answered the questions regarding do you see initial growth for leaves and do you see leaves, which mean the fully unfolded, basically adult-sized leaves, then you would be prompted with, do you see flowers or flower buds? And if you said yes, you would then be prompted with how many or how much, how many, and then you could answer, do I see less than 3 flowers open? Do I see between 3 and 10 flowers open, or more than that? And there are about 5 categories there. And so this is a quick snapshot of what the data sheet looks like. Once you get familiar with the descriptions for each of those events, which we call phenophases, you're simply going outside and looking at your plant, and you would say, log the date, and look at your plant and answer, do I see initial growth? And you'd answer yes or no. Or if you're not certain, we offer the opportunity for folks to circle a question mark so that they're still able to submit data, but they're not, they're discouraged from logging data that are inaccurate. And then the line here represents the intensity or the abundance information. And so each time you go out, you are invited to answer yes or no to all of those different questions. And so a column would represent all of the observations that would be made on a single plant on a single date. This program's been, excuse me, active since 2009. And since that date, we've amassed almost 22 million records coming from about 14,000 sites submitted by 18,000 observers across the country. And this gives you a quick sense of the distribution and the density of those records. You'll see that in certain parts of the country, we have much more data and observers than in others, and this generally follows population density. And so we are working actively to try to encourage richer and deeper data collection in some of the areas where we have not as much. But one of the things that we've really tried to focus on is that it's an easy program to get started on. And so this is a screen capture from our website. We advertise it as it's basically 3 easy steps. You simply join the program, which is meaning that you create an account, and that just requires an email account. Then you set up your account, meaning you select which species you're going to monitor. You register a site, which is usually your yard or a research plot. And then you also register the individual plants and animals, and then you begin observing. And so this is a screen capture of what we call our observer dashboard. This is mine. And so when I log into my account on the computer, this is what I see. And so on the left is the list of sites that I have registered where I take observations. What this is showing is a list of the plants and animals that were registered at a particular site. You can get more information and data sheets for an individual plant from this location. And then finally, from this interface, you can readily move on to where you enter the data. You can download the data as an Excel file, and you can visualize the data. I'll show in a moment that we have an online visualization tool where you can explore patterns very readily in the data. And this link right here makes it very easy to explore the patterns in the data that you have collected. One of the things that we do to encourage data on particular species is to run what we call campaigns. And we typically develop these campaigns in collaboration with researchers or managers. So going back to the very beginning of the workflow, where we showed, we first identify with an end user what data they need. Those needs tend to drive the focus of the campaigns. And so, for example, the Nectar Connectors campaign is one that through which we encourage observers to track the timing of flowering in a lot of plants that are important to migrating butterflies like monarch butterflies, so that we can better understand whether the plants are flowering when the monarchs are present over the course of their migration. And so each of these campaigns is helping us collect data that will directly answer questions that researchers or managers have about when things are occurring in their area. I wanted to mention this too, since this may be helpful to you if you're considering launching a program. When we create a campaign, we create what we call a landing page, a page on our website that gives a lot of information about that particular campaign. And so on the left here, this is a screen capture from our website that describes one of the campaigns in detail, one that we just launched called the Pesky Plant Trackers. It focuses on 2 different species of aggressive invasive plants in the middle part of our country. And so this website describes why we have this campaign, why we want observations on these plants, and then how to get started. And so the screen cap on the right is just what is the rest of the content further down on the page, and it walks someone through how to participate in this campaign. And so if you are an individual who has never heard of Nature's Notebook before, but someone suggests you might want to participate in the Pesky Plant Trackers program, they can come directly to this page and it will walk them through how to set up the account, how to get the proper information to get going, and then how to participate. Then one of the things that we do is once individuals sign up and participate in campaigns, we send them periodic emails that give them updates about the campaigns. And typically these are monthly or every 6 weeks or so. And so this is an example of a campaign message that we just sent out to folks participating in our, what we call our Green Wave campaign, that focuses on maples, oaks, and poplars, or acers, quercus, and populus species. And what you'll see is that we summarize information about what people are reporting and across the country, as well as what the data seem to indicate, when are things occurring based on the information that people are submitting. And this form of reporting back to participants seems very important for keeping them interested and committed and continuing to participate. So we do invest quite a bit of energy in making sure that we are reporting back to our participants regularly in this way. And since we are a small staff of about 10 people located mostly in the Southwest in Tucson, Arizona, and we're trying to interact with individuals across the whole country, we have to rely heavily on communicating using our website and email newsletters such as this and social media. It's impossible for us to go give presentations in person to everybody. So we invite anyone at all who's interested in participating in Nature's Notebook to do so, and anybody can go to the website and sign up and get started. One of the things that we cultivate quite a bit is that we have the ability for individuals to participate as part of a group as well. And so in the States, it's common for groups of individuals such as students in a class to participate together and all track the phenology of plants in their schoolyard as a team. We use that same model at some of our managed lands, like at our national parks or our national wildlife refuges across the country, as well as at a lot of colleges and universities. And so over the years, we've grown to over 500 of those partner groups contributing data to Nature's Notebook. And we've recognized that this is a really important model to cultivate because the groups tend to participate longer for more years generally than individual observers tend to, and they typically submit more data. We end up getting more information about them. And so we put a lot of emphasis on supporting groups of this sort and providing formal training for them so that it is easy for them to hopefully continue participating with us in this way. And this is a graph that just shows pretty quickly that the data that are coming in from individuals, individual participants, which is the blue line, has grown over the years, and that the amount of data that's coming in from members of those partner groups has also grown at a higher rate. In the last few years, things have kind of leveled off, but you can see very clearly from this graph that it's worth us continuing to support those groups because they are yielding nearly double the amount of data as the individual observers are. Hopefully you can see this. This is an animation that we captured demonstrating the use of one of our access tools. We have a site called what we call our Phenology Observation Portal. Oh, it's a little mushed here. This is the URL for it. All of the data that are submitted to Nature's Notebook can be freely and readily accessed in a whole variety of ways. One of the easiest ways to do this is to use this Phenology observation portal, where, as the animation is demonstrating, you can constrain and query based on the date range, the geography, the species, and even the phenophase. If you're interested, say, only in flowering phenology, you can constrain what you download to just flowering phenology. And then you simply download the data as a Excel file, and you can bring it into your statistics software or Excel for further manipulation. And it works really quite smoothly. This is another animation that I hope is coming across okay. This is just a small demonstration of the visualization tool that we have online. And if you're interested in how this works, I encourage you to just log on and check it out yourself, because anybody can access this. You do not need to have a Nature's Notebook account to access any of the data at all. The URL here is at the top. It's USA-NPN.org. Just got covered up so I can't see it, /data/visualizations. And what you can do using this tool is plot the data very readily to compare across sites, across years, and in the case of the graph that's being generated right now in the animation, across species. And what this plot is showing is that in 2019, when monarchs were flying, which is the orange line, was coincident with when common milkweed had open flowers, which was the green line. To move on now, we had just spoken about how we collect the data in order to address the needs of our end users. Once we have the data, then we try to develop models. And this is something that generally we work with external collaborators on. But I can give an example here of one of the models that we have operationalized into the form of products that have become really quite popular here in this country. So a lot of spring activity, especially in plants, but in animals too, it occurs as the result of increasingly warmer temperatures in the spring. And so we can generally envision this as if we have the date going across the x-axis, beginning at the start of the year, and then the y-axis representing accumulated warmth, meaning temperature above a certain base temperature, typically freezing. We can keep track of how much warmth a plant is exposed to since the start of the year, or typically since winter. And in many cases, it's fairly easy to pinpoint, it's possible anyways, to pinpoint how much warmth needs to accumulate to trigger something like leaf out. And generally, we can then, oh shoot, sorry. We can also do that for things like flowering. And so here in the States, a researcher at the University of Wisconsin-Milwaukee was able to establish that leaf out in lilacs and honeysuckles occurs after a certain amount of warmth, and then the flowering in those species occurs after a bit more warmth has accumulated, and that those events generally represent the beginning of biological springtime activity in plants and animals among species that undergo their activity the earliest. And so what we have done is taken those models and are applying them to daily maps of temperature, minimum and maximum temperature, and are able to produce real-time and short-term forecasts indicating the timing of spring biological activity in the form of products. And so this map is an example of that kind of product that we are using, leveraging that model. And so this map is showing for the conterminous United States, what day of year this year in 2020 did we see the beginning of biological activity? So when was that leaf index reached? And it's showing you, as you probably aren't surprised to see, that spring began the earliest at the most southern locations and then progressed northward as warmth accumulated over the course of the spring season. It gets messier in the western US because we have more topography out there, and that interacts with latitude to create more complex maps. But generally you're seeing that spring occurs earliest in the southern locations and low latitudes. What we can do is then compare when spring occurred this year to when it normally occurs and when it has occurred in the last several decades. And so this map is showing the difference between when spring leaf out occurred this year as compared to when it typically has occurred in the past. And we call that an anomaly. And what you see is that in the red tones, that's where spring occurred earlier this year. And the blue tones indicate where this year springtime occurred later than average. And so what we had occurred this past year was that the spring started extremely early, in some cases 2 and 3 weeks early, across much of the south and southeast part of the country. And then it really slowed down. And in much of the middle part of the country, things were pretty average in terms of when leaves started coming out. And then in the upper Midwest and upper Great Plains area and the Northeast, we are actually seeing a pretty late start to spring. And so these kinds of things are helpful for planners and decision makers to try and anticipate when things might occur this year. These maps have been used quite extensively in our country to understand things like, and anticipate things like whether allergies are starting earlier than average. This image on the left was from some news coverage in the Southeast where we were experiencing very early start of spring, and we also were seeing very early start of pollen release, and therefore allergies and allergy sufferers definitely could relate to that. And then hearkening back to the cultural events, we have a very famous cherry blossom festival at our nation's capital in Washington, D.C. And there was a lot of concern this year that these cherry blossoms were going to flower in advance of the festival that was planned for mid-March and early April. And indeed, the flowers did open a bit earlier. They ended up not having the festival because of coronavirus, unfortunately, but there was a lot of interest in this among the media up until that point. And then finally, our maps are referenced quite a bit by folks that work with the agricultural industry. This is a tweet where a person who works closely with agricultural specialists in the middle of the country said, if we're having an early spring, that could be bad for some of our crop species, because if they initiate their growth early in the year and then are hit with a late season frost, that can be very damaging. And there have been incidents in recent years where fruit crops in particular, cherries and peaches and other fruit trees have been devastated because of an early start to the season and then a frost that comes and decimates the blossoms, and therefore they can't produce fruit. Another couple of set of maps that we make that are similar to the ones we just saw is simply show the accumulation of warmth, again, since the start of the year on the left, and then again, anomaly or difference from average, difference from what is normal, shown on the right. And again, those maps are, that map in particular on the right is very consistent with what we saw in the spring index, where warmth is very far ahead of schedule in the south and the east, and then behind schedule up in the upper Midwest, which again has consequences for when things will occur. And one of the things that you can do using our online visualization tool is to drop a pin on a particular location and then look at that accumulation curve. And so this was from a location in the northeast part of the country from this year. And what this is showing is that the date on the x-axis starting from January 1st, and then that accumulation of warmth on the y-axis. The black line is showing the long-term average, or how the rate at which heat usually occurs or has in the past. And then the blue line is this year. And so you can see that at this particular location this year, heat accumulation was pretty far ahead of what's normal in the middle of March. Whoops. And then it drifted back a little bit to just a couple of days ago when I pulled this plot. We were only 4 days ahead of schedule in terms of heat accumulation at this particular location. And then the orange line is what happened last year. And so in some cases, that's very helpful for managers to be able to compare how is this year unfolding and how does that compare to last year, because that enables managers to be able to say, well, I undertook my management, you know, on March 1st last year, but if we're ahead of schedule this year, maybe I should back things up and plan to undertake this management like herbicide treatment or planting or something earlier this year. One other thing that we have done with those heat accumulation data is to produce these maps indicating when you might actually want to take action. And so in this case, we are predicting when you might want to control an invasive insect. Here in the United States, there's an insect called the emerald ash borer, which is a really problematic insect for ash trees, for Fraxinus. They're beautiful green emerald beetles that their larvae bore into the bark, and then they spend most of their life cycle under the bark of the tree, eating away at the heartwood and unfortunately typically killing the tree. And so it makes it hard to control them because they are underneath the bark. However, when they develop as adults, then they emerge from the tree and start flying around. And there's about 2 weeks after they emerge before they start to lay eggs on new trees and thereby spreading and laying eggs and infecting new trees. And so anticipating when those adults might be emerging is very important for arborists and tree care specialists for trying to control the spread. That's a good time to try to trap the insects. It's been established that the adults emerge typically around 450 growing degree days, or at a particular amount of warmth that has been accumulated. And so using those heat accumulation maps that we produce on a daily basis, we are also able to make these maps that indicate when you might expect those adult emerald ash borers to be flying, and therefore when you might want to take action. And so in the case of this map, which was actually back from the middle of March, the whole zone that's shown in yellow was where adult emerald ash borers were anticipated to be emerging at that time. And those were the locations that tree care specialists should be taking control. And then folks that were in the green zones would be getting ready to see the adults emerging. And so managers can track these maps and try to anticipate where their location is at relative to when they might need to control for the insects. And what we allow folks to do is register their location and sign up to receive notifications. And so in this case, a person in North Carolina signed up and indicated they would care to know when emerald ash borers were getting close to emerging. And they would be sent an email saying, okay, in this case, adults are expected in 2 weeks. And so hopefully that gives folks a little bit more opportunity to prepare and anticipate when they would need to be out there treating or trapping. Okay, so just a little bit too, that's a lot about the data and how we make the stuff we make, the information and the data products. These data have been used pretty widely. These data infrastructure and resources have been really pretty widely used by a lot of external researchers and partners. And so I will share just a few examples of how these data have been used to advance science and our general understanding of phenology. We've had over almost 90 publications now, research papers that have analyzed the data in one way or another. Typically, these fall into 3 broad categories. On the left, we see papers that are basically analyzing the data to try to understand to a greater degree, what are the conditions that drive events like leaf out or flowering or leaf color change in a particular species or in a particular location, because the drivers can vary across, say, agroclimatic zones. We also see people using the data to understand how things have changed, how the timing of a particular event may have advanced. And then finally, our data are used quite a bit to validate information that's collected by satellites and on planes and other forms of remote sensing. This is an example of where a researcher has taken our observations and built forecast models and is generating forecasts all on their own outside of our involvement. And that is just a strong example of the fact that we have sufficient data and it is of sufficient quality to be used in this way. Another recent paper that is really very fascinating is an example where our data have been combined with phenology data that were collected all across Europe. And what the researchers did here was look at the timing of leaf out and flowering across a latitudinal gradient, as well as the timing in rural versus urban locations. It's been documented already that the phenology of plants tends to be advanced in urban areas because urban areas tend to be warmer. This is called the urban heat island effect. But the researchers were curious to know whether that pattern was consistent across latitude. And what they found was that indeed at northern latitudes where conditions are temperature limited, you do see earlier leaf out or flowering in urban areas compared to rural areas at the same latitude. However, once you get down to the deep south, the latitudes at the very bottom of the countries, the relationship actually turns inside out, and the phenology at rural areas is actually more advanced than in urban areas. And so that was published just recently in Nature, Ecology, and Evolution. And then finally, this is just one example of many papers that have used our data to evaluate what the drivers of phenology are. In this particular study, the researchers compared observational data collected through Nature's Notebook to thousands of records that had been collected in previous decades and were stored as herbarium records. And through a pretty sophisticated statistical analysis, we're able to identify that it's not just accumulated warmth or temperature in the springtime that influences the timing of flowering. It's actually a number of other interesting variables. It's things like, and these are abbreviated, this is not easy to understand, number of frost-free days, or the amount of precipitation, and even the amount of precipitation as snow. And so this paper was fascinating because no one had ever really identified that variables from the winter time, things indicating the amount of snow or the timing of frost, really had that much importance on flower development and flower opening timing, but this suggests that it indeed does. And then finally, this is a study that was undertaken by a graduate student, and she worked with us. This was one of the campaigns that we ran. For several years, we encouraged participants in Nature's Notebook to keep track of when leaves came out and when leaves changed color in both native shrubs and invasive shrubs. And we had over 1,000 participants contribute data to her project. And what she was able to show was that in this plot, we see the latitude on the x-axis and the date of leaf out on the y-axis. And what she saw very clearly was that leaf out occurred in the invasive species much earlier than in the native species, but that the further north you went, the less that relationship existed. It was a very strong difference at low latitudes, and the further north you go, the less that pattern is apparent. And then finally, this is an example of one of our land management agencies, the US Fish and Wildlife Service. They use Nature's Notebook and the phenology data that are collected through the program to better understand when they might undertake activities like when to treat invasive plants. This image is from an island in the Pacific Ocean, and it is inundated by a weed called verbicina. And they needed to know when they should when they should treat, because you want to pull it before it goes to seed, because if the plant is in seed when you're pulling it, then the seeds tend to disperse and you are not controlling the plant. And so they took observations of these plants and when they were flowering over several years, and they kept track in particular of when the fruits were ripe. And they were able to identify that the fruits occurred at a pretty clear window, and that there were gaps in when those fruits were present, and that those tended to occur at similar times of year each year. And so they're now using this information to time when they should have volunteers out there pulling the shrub and trying to control it, and are actually being successful at controlling the plant on this island. And then finally, as Dr. Garcotti mentioned in my introduction, we've had our program featured on several broadcasts, national scale television and radio broadcasts, which has been really great for helping spread the word about what phenology is, what Nature's Notebook is, and why it's important. And that it's been a really great way to engage the general public, because, like I said before, everybody, even if they don't know the word phenology, they definitely notice when flowers come out and when birds arrive in the spring. And it's a way to have a conversation that enables you to better communicate scientific concepts pretty readily to folks who may not have necessarily been interested. And also, we are working hard to try and ensure that everything that we do also supports broadly folks from all different backgrounds and cultures across our country. And with that, I think I will stop and take questions. Hopefully we're doing okay on time. Thank you so much. So how should we do this? I see an awful lot of questions on there. Sarah, are you? Let's see. Sarah, are you comfortable? Have you been tracking the questions, and are you able to pitch me some?
Yeah, it doesn't seem like there have been very many questions in the chat.
Okay.
So if anybody has them, feel free to type some now. Thank you all again for checking in. We have a couple that say excellent presentation, so thank you again, Theresa. Again, if you don't have the chat window open, it should be at the bottom of your Zoom window. I see there are some hands raised, so if you would like to put those in your questions in the chat box.
There's one. Sure, it looks like folks are raising their hand. So, all right, Aditi, I'm going to open it up for you. I've just unmuted you.
Hi, am I audible?
Hi, yes.
Yeah, so I'm Aditi Mishra and I am a student of MSc in Environment Management, and my dissertation, master's dissertation, was on plant phenology, where I was using these phenocams or digital time-lapse cameras. But most of my plants were spring flowering plants, and this COVID-19 just showed up, so everything had to be left in the middle. And I'd like to ask you, like, what do you think is the most reliable method for this data collection in like visual observation or these digital time-lapse cameras or remote sensing? And can you just tell us something about the monitoring phenology using remote sensing? And another thing, like, do you think that there has not been enough research carried out in India on phenology, and there needs to be an advancement in that? That's it. Thank you.
Sure. So to answer your second question first, I would say definitely there could be lots more data collected in India. I think there are so many countries where, and the United States included, the more data we can get, the better we can understand what's going on, really. And so absolutely, yes. And if anyone needs a letter of support saying that, I'm happy to provide that. Regarding tracking phenology using remote sensing techniques, that's a really great question too. I've had some experience with the phenocams and the repeat photography kind of way of tracking things. For those of you that aren't familiar, phenocams are cameras that are mounted typically on towers, oriented toward the landscape, and take photos typically on an hourly time step, I think. They typically just use a high resolution digital camera that have the red, green, and blue channels, and then those can be subsequently analyzed to evaluate things like greenness, so you can get a really good sense of the timing of green up and green down for the piece of the landscape that is shown through the view. And that tends to be a really valuable piece of information because you can typically identify what the actual individual trees are that are in the frame of view and the different species, which is different than what you can get from satellites generally. Generally, what folks tend to analyze are data from, it may be different in your country, but at least in the US, it's the MODIS platform. It's satellites based on the MODIS, or sensors on the MODIS or satellite. And Landsat and a couple of others, and those are at they're fairly large scale pixels. MODIS is 250 meters on a side and Landsat is 30 meters on a side, and so you can't identify the individual plants in there at all, and it's tricky to even know the individual species. And so you can look at the curve of greenness when it's increasing and decreasing, but it's difficult to tease apart what is responsible for that curve. And so that is where the ground-based observations that are coming in through Nature's Notebook are so helpful. It helps analysts to understand what is responsible for the greenness that they're seeing in the remote sensing information. And then the imagery from the phenocams can help link that together as a third bit of information. And so I'm not actively, our team is somewhat involved in some of those activities, but there are a lot of different research groups across the country and really globally that are looking at how to evaluate those different data channels together in conjunction and there's a lot of active research occurring. I hope that helps. All right, I've seen a few more things come through. Let's see, Sarah, do you have a suggestion on what order to go in here? Do I need to unmute you? Let's see.
There are both question and answers in the question and answers tab and some in the chat. So I will go through the chat first. We will be posting the recording link so you can watch this again or share. Do you think the ecological niche modeling data would be used for deciphering certain critical missing links to the phenological data?
That is a great question too. Yeah, so my understanding of ecological niche modeling is that you are trying to disentangle the driver, what are the conditions that define where a species is found and where its suitable habitat might be and where its real habitat, actual habitat might be. And definitely, I think that phenology intersects with that, and there's been research that's taken place over the course of several decades that have looked at the intersection between the distribution and the distribution in a species and the timing of its phenology, primarily because as conditions change over geography, that can limit where a species can be found. So things like particular plants may only grow to a certain elevation because above that elevation, it's too cold, you know, that it may have too many days of freezing at night and the plant just can't tolerate it. I think that it's really valuable to consider that because as conditions are changing with global warming, that we could start to see shifts in those limits that are changing the tolerance of species, and that can certainly impact its distribution as well as its phenology, you know, and they can interact. And so it's an area that's ripe, I think, for further research. There are some neat papers that have looked at the intersection of those two things recently, and I'm thinking of a researcher by the last name of Prevey, P-R-E-V-E-Y, that looks at both distribution and phenology and the drivers of them that are really cool and maybe worth looking into if that's your area of interest.
We've got another question here, and I am trying to go through these. How tree phenology can be correlated with agricultural crop management?
Yeah, I'm thinking maybe the application there is that if you understand the relationship between them, then you could continue to track tree phenology and use it to guide agricultural activities like planting or harvesting. I think what you need to do there is simply track the phenology of both things and look for correlation. Or if you are able to establish what the drivers are to the phenological events in the two different things, if you identify that there are similar, if they share drivers, like again, accumulated warmth, then yeah, you may be able to look at patterns in one thing and infer what the timing of a different event might be. I'd have to think for a minute of whether that very thing has been done. Yeah, I'm not sure of a great example of it, but I do think that there's a lot of potential there, and that I guess what I would say there is that's resonant with what we are doing here in the States with the National Phenology Network of trying to make the data that are incoming as relevant and useful as possible to not only researchers, but also decision makers using it in planning in the year.
We do have quite a few questions coming in, Theresa. So another one we have, observation of spring phenology seems easy to record. What about autumn phenology, which is a little tough? What is your take?
Totally agree. That's why I talked about spring. There have been far more studies that have evaluated springtime phenology and patterns and how it's changing and what the drivers are, because it is just clear and easy, I think, especially in temperate systems. And that autumn phenology, leaf color change and leaf drop and senescence are messier. It seems from the literature, the smaller amount of studies that have focused on autumn phenology, is that the drivers are harder to pinpoint and they're not as consistent across species as they are in spring. In springtime, I'm going to very generalize things, but very much in general, in spring and in temperate systems, water limited systems are a different story, but in temperate systems, accumulated warmth, warming up in the spring, can do a really pretty good job at predicting when things will occur. In the fall, we are really trying to disentangle it still, and it seems like it tends to be a mix of accumulated chill, so the inverse of accumulated warmth, but after, say, the July 1st in the Northern Hemisphere, how much do we start experiencing cool temperatures? It can also be nighttime frosts. It can be a change in sun angle, getting to shorter days. It can be the dry down that tends to occur in some regions in the later summer and fall. But it does not seem like plants, and I don't even know anything about animals here, but in particular for plants, that they don't respond to the same extent and in the same way. And so we are definitely actively trying to come up with models that would enable us to make maps showing the autumn phenology the way that we do for spring. And we're hoping to have some simple maps of that sort in time for fall this year. But because it's so much less well documented, it's just a trickier thing to do.
We also have a couple questions coming in regarding tree ring data. How can we relate phenology data with tree ring data?
Oh, I don't know a lot about that. I have talked just a couple of times. We have a fabulous tree ring laboratory of tree ring research here at the University of Arizona. And I have spoken with some researchers there about putting dendrometers onto plants, onto trees, because they're basically like belts you put on a tree, and it can keep track of the sap flow. And so over the course of the season, and I think even daily, trees actually get fatter and skinnier based on how much moisture is in them, and that there's a seasonal pattern to that, and therefore it is a form of phenology. But we haven't initiated true trekking with that just yet. I think that if we could, though, it would enable us to leverage the tree ring data, which tend to go back in time hundreds and sometimes thousands of years, and it would be fascinating to see if we could identify how phenology has changed using that particular line of evidence. But I'm not aware that anybody has even embarked on that at all. So what a cool idea, honestly.
Another question, how does phenology help in monitoring of cultural events, as you mentioned in the very beginning?
Yeah, so what I really mean there is that if the timing of a particular event, like when tulips are flowering, in the case of the tulip festivals here, if that is changing, then the organizers of the cultural event might wish to consider changing the timing of the program. For example, in a town where my parents used to live in the northern United States, they had an event called Tulip Time, which was a 2-week window where thousands and thousands of tulips were planted around the city, and they would flower, and it was a huge tourism activity and brought tons of people into the city for the event to see the tulips, and how to you know, people would spend a lot of money to be present for that event. But while during the several summers that I was there, I saw the tulips in flower completely coincident with the 2-week basically party that the city threw. And there were definitely years where the tulips flowered far in advance of the event, and by the time the event occurred, we all joked it was stem time, because all that was remaining were these stems sticking up with leaves. And so I think that there's an opportunity there maybe to better understand how much the timing of the event is varying from year to year, as well as what the drivers might be, to try and understand if it may be occurring earlier or later in the coming year, to try to help support event planners in ensuring that when they have the party, when they have the event, it actually coincides with the flowering or whatever the event might be.
And I do apologize, there are quite a few questions, so we'll try and get through some more. Another question, are you also working on seed dispersal and germination phenology?
Not really. Well, part of the protocols, going back to where you are prompted to make the answer to answer yes or no to the questions for a particular species. I didn't get down to the seed ripening and seed dispersal, but those are part of the protocol. For every plant where you could observe it, we do ask for folks to identify when seeds seem to be ripening, when they are dispersing. We call it recent seed drop, and how many or how much. And so again, it's those categories of typically things like, you know, less than 3 to 10, 11 to 100, 101 to 1,000. And so it's to try to give a sense of when is seed dispersal occurring, as well as the abundance of that thing occurring. So I guess, yes, I guess we are tracking it. I am not aware of anybody analyzing those data at this time. So there's a great opportunity, especially if someone on here is looking for a project. There is so much opportunity to evaluate our data. So many questions yet to be explored that I definitely invite it. Thank you. I see Ellen is putting in a bit of information in there too, and I appreciate it. Ellen Denny is our the individual on our team who has developed all of the protocols, and she's on here too. So she'll help keep me honest on these things. Sarah, you got another one?
Yeah, there's another question. How can we link phenology and food security?
That's a tough one. Again, I think I'm going to say there's an awful lot of opportunity there. At the minimum, we can look at historical data. Again, the timing of when crops were planted and when they came to market, when they were ready for harvest, and look to see how that might be changing. And again, try to pinpoint what the driving conditions underlying that development might be. And I am not an expert in agriculture by any means, but we definitely are familiar with the fact that that phenology is used very, very extensively in agriculture. And so there's a lot of opportunity there for understanding when to plant and what the development cycle is and how that might be changing. And then another really important piece to the puzzle is evolving mismatches between plants that need to be pollinated and the availability of pollinators. And with the huge drop that we're seeing in honeybees and their importance, it's a pretty important, pretty serious concern, honestly, about food security. And so being able to better understand how the timing of flowering in particular crops may be changing, as well as the presence and abundance of necessary pollinating organisms is critical. That's not research that our office has undertaken, but again, I think that there's a great deal of opportunity, and we would be really keen to work with folks that were interested in pursuing that further.
Another question, how can we calculate the base temperature for leafing from species phenology?
Ah, yes, okay. If whoever put that question in, if you email me, I would be happy to send you some literature on this. It's typically a regression. What I have seen, how I have seen folks, researchers have done this is using regression, and just it's kind of trial and error, honestly, where different base temperatures are iteratively tried out with different subsets of data to look at model fit and performance. And so I think that exercise of trying to identify the optimal base temperature for predicting something is undertaken typically where there's a lot of observations that have been collected at a single location, so that you're removing the site-to-site variation. In a lot of other applications, I have seen that a more generalizable model that can be used across geography typically just uses 0 centigrade or 10 centigrade or Celsius as the base temperature. Those are fairly common base temperatures that are involved in a lot of predictive models of heat accumulation, and seems to work well enough in a lot of applications. But yeah, please do follow up with me. My email is on the screen if you want to see the papers about how folks have experimented with trying to pinpoint a particular base temperature.
There's another question about how did you create the baseline to detect the change as well?
I'm sorry, can you say that again? I was distracted by the chat.
There's another question on how did you create the baseline to detect the change?
Oh, yes. Okay. So I'm guessing you might be referring to the anomaly map that I showed that was red and green, or sorry, red and blue for the spring index. But this is true for anything. What we use is what is called the climate normal, which is a 30-year average that takes the most recent 3 full decades. And so right now that is 1981 through 2010. But once this year, 2020, is complete, the climate normal will shift to be 1991 through 2020. And that will be what we use. We didn't come up with that. That's a fairly standard approach in the climate world. And so we just adopted that because it's a common way of calculating anomalies. However, I'm guessing what you might be getting at is that you can get really different answers depending on what you're comparing your current year data to as your baseline. And I'm anticipating that, yes, once we update the climate normals to the most recent 3 decades, we'll start to see really different things because there is that underlying increase in temperatures in recent years that will make the differences from normal be different. So hopefully that answers the question.
Another question we've got, whether the predictions for the phenological changes are made based on the data collected from the same plants marked and recorded over years, or is it a random selection every year? And I also, the second part of the question is, could you please let us know about the phenology protocol? I put the link for our report on our protocols in the chat, so you can check out that page.
So for the first part, asking about which data are we using to build models, so far what we've done is use historical data that have been collected, and we construct a best fit and then have operationalized that. You're right, though, that there's absolutely the opportunity to on an annual basis or even more frequently than that, evaluate the model performance with incoming data. And we hope to do that. We just aren't to that level of sophistication just yet. There's nothing to prevent anybody else, though, that are using our data to do that. And I think that some of our data are being used in iterative forecasts of that sort, where models are constructed and as data are submitted, the model is reevaluated against those data and perhaps updated. And so that we're looking to some of those research activities that are being carried out by other parties, by other researchers, to help us better understand how we might operationalize that at some point in the future once our capabilities permit that.
All right, next question. Crucial precautions we must take before tagging a tree.
Oh, that's a good thought. I'm gonna, Ellen, you're on here. Tell me if I forget anything. For sure, we strongly encourage that you get permission to access the property if it's not your property. And so, especially here, we have a lot of people observing on public lands like national parks or even private parks. And generally those sites are accessible because they're public, but for example, right now, a lot of them are closed with the shutdowns. And so we're strongly encouraging our observers, don't go where it's not safe to go right now, either because the property is closed or because of potential exposure to the virus. We also recommend that you tag it in a way that it's going to be permanent enough that you can find it again. And sometimes you need to refresh the tag that you have on there if it's getting tattered or worn. But ideally, yes, you want to tag it so that you can find it again, but in a way that doesn't damage the plant in any way. So we tend to encourage stakes that you can put in the ground, or flags or tags that you can put around the base of the tree, but that wouldn't girdle it or cut into it. We discourage poking it or putting nails or anything like that in, because that can cause damage or introduce disease. And Sarah, would you like to speak to that? Sarah has actually made a ton of plant tags and distributed them over the years. So maybe you have some thoughts on that.
Yeah, we use everything from small aluminum plant tags that you're able to write the plant information on one side. We've used, like Theresa said, larger stakes in the ground, some more permanent signage for individuals. We also have groups and places, locations that will use a large sign that identify all of the plants that are under observation, or use things such as a Google map where you can drop pins on the exact location coordinates of each of the individuals that are under observation.
Thank you, Sarah.
All right, I'm trying to get through the questions. Another one we've got here. The maps you showed must be interpolated from the in situ data, agreement with the satellite derived phenology. Is there any assessment done of this kind? Are you looking for both onset and offset of phenology while collecting such data? Any comparison with satellite derived products? Sorry, that's a lot. If you want me to repeat any of it, let me know.
I'm going to start with what I can remember. So the first one, actually, the maps are not interpolated from the observational data. The continuous maps that we have right now are produced using the models that have been established between the timing of the phenology and the drivers. And in the case right now of the spring index and other things, it's almost solely a function of daily minimum and max temperature. And so we get that interpolated temperature data product from another provider, various other providers here in the States. So we're not doing any interpolation yet. We bring in those temperature data, we run the model on it and produce the map. However, we are then comparing those forecasts and predictions with the observational data. Not a lot of that is actually available on the website just yet because it's, for the most part, still in pilot phase. But yeah, that we're definitely trying to move into that direction much more in the coming years, where we're making predictions using models and then comparing it with the incoming data. And then, like I mentioned a little bit previously, ideally then the model may even be updated iteratively as a result of the incoming data to make it perform better in different parts of the country or at different times of the year. Right now, we're not taking advantage of remote sensing information very much in that way either. We have some remotely sensed imagery or information products on our website at the present, but we're just beginning really to be able to assimilate those various streams of data and use them in conjunction. So that's another area that's ripe for a lot of growth. And the research community has been moving very rapidly in that direction. And we're hoping to kind of learn what we can from what others in the research community are doing, and then determine what's the best fit for our capacity here, and what is of the most value for our end users. Did I get everything, Sarah? Reasonably well?
Yes. Another one, does previous year's precipitation alter the phenological events, particularly in tropical conditions?
Probably. I don't know. Yeah, I don't know a ton about tropical phenology. I know that's a much trickier region to make predictions, because if you don't have distinct seasons, it's just hard to know, which months or days or years to even consider as potential drivers. I have seen some cool literature on that part of the for that region, but I don't know.
We have a couple species, a couple questions on this. How phenology data can be used for monitoring invasives?
Yes, definitely. There's a lot of value to tracking the phenology of invasives, especially for understanding the potential for further invasion. Definitely, it's been documented that invasive species tend to be so successful in their invasions because often they have very rapid response, and their phenology enables them to, for example, put on leaves earlier in the season than the natives, and therefore they can grow more rapidly, take advantage of the resources in the soil, perhaps shade out the competitors, the native species, and then it becomes this positive feedback where they're the plants that are growing the strongest, producing a lot of seeds, and then continuing to dominate a system. And so there are definitely researchers that are using the Nature's Notebook system to understand this even further about how the phenology of invasives is different from natives, and how that confers benefits to them in ecosystems. And so, yeah, I think it's that's an area that there has been a fair amount of literature published on, and also even further opportunity to use our data that we have already to dig into it and compare natives and invasive phenology, as well as follow the example of others and collect further data on native or invasive species and better understand the differences.
Another question. Understanding phenology as far as leafing is concerned is easier in deciduous species, but can it be confusing for evergreens?
Oh man, again, Ellen is on the line. She might have things to say. For evergreens, we do encourage observers to track what is possible to be observed. And so evergreens do put on new young leaves at various times of the year. And so that is the primary phenophase that we are asking folks to observe on. But you're right, it does take a keen observer to witness that and capture it, because it's not as obvious and dramatic as with deciduous trees. Similarly, we still ask folks to observe on all of the other events like flowering and seed development and seed drop for the evergreens as well.
All right, a few more questions. I'm trying to get through
So
everything.
probably we may restrict to 1 or 2 questions now. So Theresa must have tired now. She's talking for last, say, about 2 hours.
It's been very enjoyable. All right, 2 more. We'll do 2 more, Sarah.
Okay, could you throw some light on how phenological fluctuations could alter the quality of the floral rewards like pollen and nectar?
Again, not really in my area of expertise, but my understanding is that if the period of development for particular plants is being shifted, that the amount or quantity of nectar or pollen may be perhaps negatively impacted. And considering too the previous question with things like invasives, if you've got increases in competition as a function of changes in phenology and introduced species, that may also have an impact on them, on the availability of those resources as well. But truthfully, I don't feel like I can say a whole lot about that one.
That's it.
I'm sure I must have missed a couple questions through the chat and the question and answers. So I apologize if your question did not get answered in this session.
Well, feel free to follow up with me via email. I'm happy to do my best to answer additional questions that way too. And it's been a true pleasure. So enjoyable to talk with you all. Thank you so much.
And again, we have recorded this session. We will be sending the link in case you need to watch again or share with colleagues or coworkers.
So thank you, Theresa, for your interesting and highly informative talk. So success of webinar can be gauged from the number of questions you have received. Still, we have the stock of questions, so and we have the time constraint. So on behalf of entire organizing team here and in University of Arizona, so I thank all the participants who have attended this webinar. So if some questions are left, so they can directly contact Theresa and her team. I must thank Sarah and Mayank in GNU and your team there who have worked ceaselessly to make this webinar a success. So see you all in next webinar whenever we'll conduct in future. Thank you so much.
Wonderful. Thank you so much, Dr. Garcotti. It was a pleasure. Take care.
Thank you all.
Bye.
Bye-bye.